Hey guys, ready for the third lecture of Unit 5. This is going to be on cell membranes, and then that's not very long, so I'll spend a little bit of time talking about the differences between prokaryotes and eukaryotes. So you probably remember phospholipids. These are these molecules that have a head on them that is all hydrophilic. It's got charges and things like that makes it attractive to water. And then it has tails that are just strictly hydrocarbons, so they are hydrophobic. And they can be straight tails, saturated fatty acid chains, or kinky tails with those unsaturated fatty acid chains that we talked about before. But these are phospholipids, and a lot of times we just draw them schematically like this, so that we can show this bilayer that they form. So you can see, like, this would be the outside of the cell, this would be the inside of the cell, and this phospholipid bilayer forms this hydrophobic boundary. So anything that is charged or polar cannot really make it through here. Some things make it through just fine. Anything that's hydrophobic will go right through here. It'll be very happy in here being hydrophobic. Gases like oxygen and nitrogen and carbon dioxide will go right through here too. But you got this phosphate plus additional attachments that are hydrophilic on the head, and then two hydrophobic fatty acid tails. And of course, they line up tail to tail because they're trying to minimize their exposure to water. So we end up with this hydrophobic boundary that's basically, we can think of it as being impermeable to anything that's hydrophilic. It's not entirely impermeable, but essentially it is. Phospholipids are constantly in motion. The membrane is fluid. Those guys are always moving around, circulating around. And there are even sometimes when they flip over. That's rare, but it can happen. But they are always in motion. It is a fluid. It's like olive oil. And that's largely because we have these kinky tails. These kinky tails keep it fluid. If the tails were saturated, and there are parts of the membrane that are like this, it would make it a lot stiffer. Those parts of the membrane are going to be a lot stiffer, a lot more dense, and a lot less fluid. We also have cholesterol, which we talked about before. It's a steroid, and it helps to keep the membrane fluid at different temperatures. At very low temperatures, this irregular shape kind of disrupts the regularity and keeps it from going solid. At very high temperatures, it's so hydrophobic that it holds these tails together. It holds all this closer together and keeps it from being disrupted easily. Makes it thicker, makes it more dense. So it's a mosaic. It is a combination of little bitty things, right? And a mosaic is like this picture. It's a bunch of little things that make a complete final product. And in this case, it's the membrane. So that membrane is a fluid mosaic. That's what we usually call the model, the fluid mosaic model. And it's basically this idea that there are all these molecules, a variety of molecules, because there's lots of proteins embedded in there too. So it's all these phospholipids plus cholesterol plus all these proteins embedded in there. And they are fluid and they are able to move around. And the membrane is made up of all those little bitty things. So we say fluid mosaic. So here you can see there's lots of phospholipids, there's lots of proteins, and some of these proteins are glycoproteins, which means they have chains of sugars on them. There are glycolipids, which are lipids with chains of sugars on them. There are proteins that go through the membrane. There are proteins that attach to the membrane but don't go all the way through. So there's a lot going on here. So it's a fluid. It's a mosaic. We call it the fluid mosaic model. Now, if you think about these proteins that are transmembrane proteins, right? They have a part out here that is in the watery exterior of the cell. And they've got a part in here that's in the watery interior of the cell. And then they've got this part here that hates water. So think about the R groups that you're going to have on these amino acids. We're going to have R groups on amino acids here, and we're going to have R groups on amino acids here. And they're going to be very different. Pause the video and see if you can answer that. So if you're looking at this, the pink ones here, these guys have to be hydrophobic or else they're going to be popped right out of that membrane. They have to be happy in there. So you're going to have hydrophobic R groups. And you can see these spirals. These are alpha helices that we have going on in here, which is that secondary structure. And when you have an alpha helix, that backbone is spiraled and all of the R groups are sticking out on the outside. So if you have a bunch of hydrophobic R groups on the outside, they're perfectly happy for that cylinder, for that spiral to be embedded in the cell membrane in that hydrophobic region. And so transmembrane proteins tend to have these alpha helices covered with hydrophobic, nonpolar R groups. And of course, the R groups in purple are going to be hydrophilic. Sometimes the same thing is done with beta-pleated sheets, which is what's shown here in this aquaporin. An aquaporin is just a channel that allows water to move either way. And these are pretty common in most cells. We have other proteins that have, like I said, alpha helices and things like that, embedded proteins that have hydrophobic R groups in that part that's embedded in the membrane. These proteins still have a very specific shape and can change their shape to make things happen. They can have all kinds of different functions, and we'll go through some of those. So sometimes these guys are transporters. We talked about channels and pumps when we did the cellular toolbox. Sometimes they're enzymes that are embedded in the membrane, and we've seen some of these before already. Receptors, we've definitely talked about, where they receive some signal, bind some ligand, and that causes them to do something inside the cell. We have these glycoproteins that have chains of sugars, and chains of sugars on the outside of cells are largely for identification. We'll talk more about that here in a second. Cell-to-cell attachments. We have proteins that keep cells from separating from each other. We have a variety of different types there. And then attaching to the cytoskeleton, and part of that is just keeping the structure and the shape of the cell the way it's meant to be. So like I said, these carbohydrates, these chains of sugars that are in the membrane, right? This is what, like, if you're type O blood or type AB blood or type A blood, whatever, the differences are the chains of sugars attached to a particular protein. So which sugars you have attached there, that's what makes you have different blood groups. The idea is that these are the molecules that identify your cells. This is the thing that your immune system is looking for. It's chains of sugars that are not yours. That's what it's looking for. It can also detect other molecules that are not yours, of course. But these are what we call antigens. These are markers on the outside of the cell that identify them. They're important for cells talking to other cells, and when we do cell communication in the next unit, we're going to see that a lot. I'm not really sure why this happened. I think at one point I said that cell membranes looked a lot like characters from Peep in the Big Wide World. If you don't know what this show is, you're missing out. It's a quality program. So membranes are not just outside the cell, they are also inside the cell. We have internal membranes in eukaryotic cells, right? We have membrane-bound organelles. These are locations where things happen, or locations where we allow things to be transferred. And a lot of times, we've talked about this a couple of times before, but we will change the amount of surface area by adding more folds in that membrane. And you can see that in mitochondria, you can see that in endoplasmic reticulum, and you can see that in the cell membrane itself. We will add folds to increase surface area for transport, or to increase, in the case of mitochondria, locations for those enzymatic pathways, those metabolic pathways like the electron transport chain, for those to happen, places for those to happen. Outside of the cell membrane, we have what's called the extracellular matrix. So animal cells typically have a whole bunch of proteins and sugars out there as a network around the outside. And in some of our tissues, it is mostly extracellular matrix. Now, these are proteins and sugar chains that are assembled by the cell. But if you think about like cartilage is almost entirely extracellular matrix made by very few cells. Bone is the same way. A lot of your connective tissue in your body is mostly extracellular matrix. It's stuff outside of cells with just a few cells to maintain it. And that's what gives your body its shape and its structure. Plant cells, their extracellular matrix is largely cell wall, right? So what they have going outside is chains of cellulose attached together with little proteins. So what that means is that the cells can't really be right up against each other because cell walls are really thick compared to the extracellular matrix in animal cells. And so cells actually end up being a pretty significant physical distance away from each other, which is why so many cells in plants have plasmodesmata. They have those channels that go through. The continuous cytoplasm from one cell to the next is pretty much the rule all throughout a plant. So we've looked at cell parts before, and I think we may have even seen this slide before. It's definitely worth reviewing. See if you can pause the video and answer all three of these. What kind of cell parts would you find in cells that have these features? Pause the video, try it. So any cell that's going to be secreting things is going to have lots of ER and Golgi, rough ER and Golgi, right? Because that's going to be secreting proteins. You're going to assemble those proteins in the ER, and then they're going to be transported to the Golgi, and then they're going to be transported to the cell membrane through vesicles, and they're going to be dumped outside of the cell. Muscle cells have to contract to move bones. You're going to have lots of cytoskeleton whenever you're moving anything. You're going to have lots of motor proteins whenever you're moving anything. And you're going to have lots and lots of mitochondria because you need huge amounts of ATP. Sebaceous glands secrete oils. So secreting oils is a little bit different. You are going to have vesicles, but you're going to have also a lot of smooth ER because that's where we synthesize lipids, and oils are lipids. All right, so we need to talk about the differences between prokaryotes and eukaryotes. So pause the video for a second and see if you can answer any of these guys. See if how many of these you can get correct. as to how they are different in prokaryotes and eukaryotes. So hopefully you remember prokaryotes, of course, no nucleus. Eukaryote has a nucleus. Prokaryo means before nucleus. Karyo is a nucleus. Eukaryo, eu means true. Karyo means nucleus. They have a nucleus. Same thing with membrane-bound organelles. There are infoldings of membrane in some bacteria, but they are not true membrane-bound organelles. They are continuous with the outside of the cell. It's just infoldings. These are much, much smaller, tiny compared to eukaryotic cells, much simpler also. So prokaryotes tend to be very, very simple. But that also means that they can reproduce much more quickly, right? They can reproduce in 20 minutes. Most bacteria in favorable circumstances can reproduce in 20 minutes. Your cells, even when you're an early embryo, they don't really, they can just about match that. But as a very early embryo, we are dividing without growing because we have the largest animal cell, which is an egg cell. It has the volume of many cells, so it actually divides several times without growing at all. So in that circumstance, it can divide very quickly. But typically, there's a lot of time in between for growth. The DNA in prokaryotes is circular. There are no ends to the chromosomes. It is generally naked with the exception of Archaebacteria. It doesn't have histone proteins or any other proteins really associated with it. Very few proteins, and really those are just regulatory proteins that are associated with the DNA. Whereas ours have histones, we have introns, which we'll talk more about later. Our DNA is used in a much more complex way, and it's much more highly regulated. The regulation system of gene expression in bacteria is much simpler. Prokaryotes do typically have a cell wall. It's made of a kind of a protein and sugar combination called peptidoglycans. And of course, some eukaryotic cells have cell walls, but they're either cellulose in plants or chitin in funguses. Prokaryotes, at least 3.5 billion years ago, pretty much as long as the Earth has been capable of producing fossils, because before that, a lot of the Earth was molten and we didn't end up keeping whatever was on the surface anyway. Eukaryotes, roughly 2 billion years ago, it's still kind of hard to pinpoint because it's the kind of thing that doesn't leave a huge amount of evidence. And when it does, we keep finding examples from different times. But they do seem to be the oldest ones around 2 billion years ago. So there was a long time on Earth when all life was prokaryotic. This is kind of a cool image that allows you to see the size difference. These are prokaryotic bacteria in your body. Well, this is probably not in your body, but this is the way it works in your body. This is a white blood cell, a macrophage cruising around and eating these guys. And we'll do endocytosis, bring those guys into this cell, and then fuse a lysosome to it to digest them. But there are things with prokaryotes and eukaryotes have in common. And structurally, that's pretty easy. I mean, they have DNA, right? And they have ribosomes. And their DNA and ribosomes work essentially exactly the same way. I mean, if I have AAA in the DNA and I go to transcribe that AAA, I'm going to make an RNA that is UUU, and every living thing on Earth does it that way. That RNA gets handed to a ribosome, and even though the ribosomes are kind of different structurally, their function is essentially the same. They're going to take UUU and they're going to add the next amino acid according to that UUU code, and that will be phenylalanine. They follow the exact same code. It doesn't matter what living thing you are on Earth, you follow that exact same code. If you think about it, that should make sense. I mean, that's evidence that this happened one time and we've stuck with this system, because if you change that, if you change what the DNA code, what amino acid that codes for, you're going to change what your protein shape is of every single protein you have. And so suddenly all the things that used to work great are no longer working at all. So we really can't change that without, being completely detrimental to that living organism. Of course, we also have, cell membrane, and it's a phospholipid bilayer. It's the same in prokaryotes and eukaryotes. We have cytoplasm, which is, the same stuff, proteins and water and some salts, sugars, things like that. So that's essentially the same stuff too. So a lot of the functions are the same, especially with this DNA ribosome business. A lot of the proteins, especially the metabolic ones, work in a very similar way. So I've mentioned before this division of domains, right, above kingdoms now, because we have bacteria that in some ways are much more similar to eukaryotes than they are to other bacteria. And so that's this Archaebacteria group. And if you look, we've split these domains in such a way that we have a common ancestor, but there is a more recent common ancestor with Archaebacteria than there is with bacteria. And the reason we say that is because a lot of our DNA looks more like archaebacteria. Archaebacteria, they use histones. Archaebacteria have introns that have to be removed from the transcripts. And I know we haven't talked about that yet, but we will, and it'll make more sense later on. But basically, the RNA, after it's made as a copy from the DNA, it gets edited. It gets parts snipped out of it. We do that. Archaebacteria do that. The rest of the prokaryotic world does not do that. Their ribosomes and their replication enzymes are a lot more like ours, but those are just genetic differences. And again, this is things we learned once we started sequencing genes and looking more closely at the actual genetic material. So bacteria absolutely live everywhere. If you wanted to pick a category of organism that's most successful on Earth, these guys vastly outnumber us. They vastly outweigh us. Every place we go, they are already there. And if they aren't already there, when we go, we're taking them with us because they're always with us. They live in and on all organisms. They live in the soil. They live in the deepest oceans, under extreme conditions, on the living, on the dead, in the living, and in the dead. These places where water is nearly boiling, this is where archaebacteria live. They are pretty diverse. They come in a lot of different shapes, and a lot of the names that you may have heard come from their shapes. Spirulina is a cyanobacteria that's spiral shaped. Strepto means they are strung together in chains. Coccus means they're essentially ball shaped, and so there's all these little balls. This is Staphylococcus. Staphylo just means clumped. Coccus means balls. There's a lot of rod-shaped bacteria like E. coli or the Bacillus bacteria. So they come in a variety of different shapes and sizes, and they do live in so many different environments. Here's a cyanobacteria. Like I said, infoldings of the membrane, lots and lots of infoldings, adding surface area because they're doing that electron transport chain. If you think about what's happening in photosynthesis, remember the light reactions happen in that thylakoid membrane, which is, those discs and just stacks and stacks of these discs, which is huge amounts of surface area. That originates with these infoldings that give surface area to a cyanobacterium. This is a photosynthetic bacterium. You can see lots and lots of infoldings of the cell membrane all through here. We have a couple of different types of cell walls. This is not something you really need to know about, but sometimes they have this double membrane in some bacteria. When we go to stain bacteria, there's a certain type of stain that will stain to this kind of bacteria, but not this one. And so we use this Gram staining technique, and this is one of the ways that we identify bacteria. The stain will stick to some of them and not to others, and so they'll show up different colors in a slide in a microscope. So bacteria divide very rapidly. Like I said, this is about 20 minutes per division, but one bacteria, with 20 minutes doubling time, in a day, there's millions and millions of them, right? Huge numbers of them. That's the power of their simple system, right? They are able to copy everything inside their cell and divide so quickly because of the simplicity of the organism. So pretty short video today. Hopefully you guys got all that. It'll be stuff we reference quite a bit later on. You need to know about cell membranes, and you need to know the differences between prokaryotes and eukaryotes. AP likes to ask about those prokaryote-eukaryote differences and the prokaryote-eukaryote similarities. So make sure you know those well. Hang in there, you're doing great.